Welcome back to the deep dive. We are looking at a stack of research today that honestly feels a bit like we're peeking behind the curtain of how nature actually works.
Yeah, it really does.
We're digging into a new paper out of MIT, Harvard Medical School, Apple, and Notre Dame. It's titled Protein Structure Tokenization via Geometric Byte Pair Encoding.
I mean, it's a dense title, I'll give you that, but
Right.
the implications are massive.
It sounds dense, yeah, but the mission here is actually pretty clean. We're talking about teaching computers to read the language of life. And I don't mean the genetic code, the A's, C's, G's, and T's. We've had that down for a while now.
Right, the sequencing is a solved problem, relatively speaking.
Exactly. What we are talking about today is reading the actual physical shape of biology.
And that distinction is everything, because for the last few years, we've seen this absolute explosion in AI for biology. We have these massive protein language models, or PLMs,
Right.
and they treat biology just like ChatGPT treats English text. They look at a sequence of letters, in this case amino acids, and they just predict the next one.
Which works great for text. But proteins aren't just text files floating in a void somewhere.
No, they are physical machines. They fold, they twist, they lock into very specific 3D configurations. If you get the sequence right but the shape wrong, the protein is utterly useless.
It's like having the blueprint for a Ferrari but building it out of wet cardboard. I mean, it's not going to drive anywhere.
That is the perfect way to look at it, and that's the disconnect we're exploring in this deep dive. Current AI is amazing at the spelling of biology, but it is really struggling with the geometry.
So this paper introduces a new system called GEOBPE that tries to fix that exact problem. But before we get to the solution, let's establish why this is so hard in the first place. Why can't we just feed 3D shapes into a computer? We do it for video games and CGI all the time.
We do, but biological data is uniquely difficult. To get an AI to understand a complex, continuous shape, you usually have to digitize it. You have to break it down into discrete chunks or tokens.
Right, make it readable for the machine.
Exactly. And the standard way the field has been doing this is with something called VQVAEs.
Vector quantized variational autoencoders.
That's the one. And without getting bogged down in the math, the underlying philosophy of a VQVAE is basically taking a complex curve, chopping it into pieces, and assigning each piece a code from a fixed codebook.
So it's like a list of arbitrary barcodes.
Yes. You're taking a unique, fluid curve of a protein backbone and forcing it to be, say, code number 452.
And that's a problem because, well, for one, it's a black box. Code 452 is just a random vector in a computer. A human expert can't look at it and say, oh, that's an alpha helix. It means absolutely nothing to us.
Right, there's no interpretability. And the second reason is that it's rigid. Biology is messy. It's incredibly noisy.
Proteins vibrate and shift constantly.
Exactly. So if you have a code book of rigid, fixed shapes and you encounter a protein that is just slightly different, maybe bent a few degrees to the left, the VQVAE struggles immensely.
It forces that unique shape into a box that just doesn't quite fit.
It's like trying to describe a cloud using only square blocks. You lose all the nuance.
And that loss of nuance has to kill your ability to generalize, right?
It completely does. When these older models see a new protein structure that isn't perfectly represented in their training data, they fail. They just don't have a barcode for it.
Okay, so that's the status quo for you listening. We have models that are rigid, they're opaque, and they struggle with the messiness of real life. Enter GEOBPE. The researchers call it a principled protein structure tokenizer. What makes it principled?
Well, they decided to stop fighting the analogy between human language and biology and instead lean all the way into it. They took a technique that is the absolute bedrock of modern language models, Byte Pair Encoding, or BPE, and applied it to physical 3D geometry.
Let's unpack BPE for a second, because that's a term we hear a lot in the AI space. In text, this is really all about efficiency.
Correct. If you feed raw text into an AI, you don't want to feed it character by single character. That is way too slow and computationally expensive.
So BPE looks for patterns.
Exactly. It scans the data. In English, the letters T and H appear together constantly. The, that, this. So the algorithm merges them into a single new token.
And then it probably sees T and E together a lot, so it merges them into the word the.
You've got it. It builds a hierarchy. It starts with atoms, the individual letters, and builds up to molecules, the words. It learns that ing is a highly useful chunk. It learns that emulsation is a chunk. It creates a whole vocabulary based purely on frequency.
So GEOBP is doing exactly this, but for protein shapes instead of text.
That is the core innovation here. Instead of letters, their atomic unit is the bond residue. These are the tiny fundamental structural units of a protein backbone.
Okay.
The algorithm scans a database of thousands of proteins and looks for pairs of these 3D shapes that just happen to appear next to each other frequently.
So it's looking for a specific twist or a specific bend that nature uses over and over again.
Yes, they call them geopairs. It finds the most common pair, merges them into a single new token, and then repeats the entire process.
Creating a merge tree.
Exactly. It starts with single residues, combines them into slightly larger motifs, and then combines those into even larger domains.
I really love the logic of this because it feels like how we build things in the physical world. It reminds me a lot of Legos.
Oh, that's a great analogy.
Yeah, think about it. You have the single one-by-one bricks, but eventually you realize you're always building this specific window frame or this specific wheel axle out of those smaller bricks.
Right.
So to save time, the factory just manufactures that complex window frame as a single ready-to-use unit.
That is a really strong visualization for what's happening. GEOBPE is essentially analyzing all the buildings of biology to figure out what the custom Lego bricks should be.
But there's always a catch with biology. Legos are perfect. They snap together flawlessly. A 2x4 brick is always exactly a 2x4 brick.
And biology is wet and wiggly.
Right. If GEOBPE finds 1,000 instances of a specific helix, they aren't going to be identical clones of each other.
You've just hit on the hardest part of this entire project. How do you define a single word when every single time nature writes it, the handwriting is slightly different?
So how do they solve the handwriting problem?
They solve it with clustering. Specifically, a technique called commodoids clustering.
Meaning they just find the average shape?
Sort of, but it's more specific than that. They look at all those 1,000 wiggly, slightly different versions of the shape, and they find the one real-world example that is the most representative prototype, the one sitting right in the center of the cluster.
Okay, but doesn't that bring us right back to the square blocks for clouds problem we talked about earlier? If I take a messy organic curve and snap it to this idealized prototype token, I'm introducing error.
You are. You absolutely are. And in 3D space, that error is dangerous. The paper refers to this as geometric drift.
Geometric drift, let's break that down for the listener.
Imagine you're building that Lego tower you mentioned, but every single brick you use is just a tiny bit warped, maybe just 0.1 degrees off.
By the time you stack 100 of them, your tower isn't just leaning.
It's completely falling over. Or the top of the tower ends up being 3 feet to the left of where it should actually be.
Right. So in a protein chain, if you just snap these rigid tokens together one after another, the accumulated error means the end of the protein might be 10 or 20 angstroms away from its correct biological position.
Exactly. The protein creates a massive structural disconnect. It literally breaks the chain.
So you have this great vocabulary, but you can't actually write a sentence with it because the words don't connect to each other.
Right. And this is where GEOBPE does something incredibly clever. It separates the what from the how.
What do you mean?
The what is the rigid token, the shape of the block itself, but the how is the connection between them. To fix the connections, they use differentiable inverse kinematics.
Now, that is a mouthful, but inverse kinematics is a robotics term, isn't it?
It is. Think about a robotic arm on an assembly line. If you want the robot's gripper to be at a very specific coordinate in space to grab a car door, you have to mathematically calculate the angles of the shoulder, the elbow, and the wrist to get it there. You solve backwards from the destination down to the joints.
So in our protein model, the joints are the connections between the tokens.
Precisely. They call them glue angles. Specifically, they are looking at the torsion angles, theta, phi, and omega.
So the algorithm keeps the token shapes totally rigid so the AI can read them like distinct words.
Yes, but it optimizes these glue angles to absorb all that accumulated error.
It wiggles the joints so the chain stays perfectly connected. That is brilliant.
It is. It mathematically optimizes them using what they call an SE3 end-frame loss.
Which basically means what in plain English?
It means the system checks where the protein chain should end in 3D space, sees where it currently does end with the rigid blocks, and adjusts those glue angles backward until those 2 points match perfectly.
That is the secret sauce right there. It allows you to have the discrete, readable tokens that computer language models need.
While using the glue to handle the continuous, messy physics that nature dictates, it's the bridge between the two worlds.
It really is. It's the first time we've seen a tokenizer that genuinely respects the continuous nature of biological physics while still giving us discrete units to compute with.
Exactly.
So mechanically, it makes total sense. But we have to ask, does it actually work better? Usually when you introduce a complex compression scheme like this into a system, you lose something in the fidelity.
You'd think so, but the stats here are honestly surprising. The first thing to look at is the efficiency. They measured bits per residue, which is basically just how much digital data you need to describe the protein.
And how did GEOBPE do?
It achieved a greater than 10x reduction compared to other leading tokenizers like ProToken.
10 times smaller. That's a massive win for storage alone. But does it actually matter for the AI's day-to-day performance?
It matters hugely for the context window. If your
Correct.
protein description is a massive file, the AI can only hold a small chunk of it in its memory at a time.
You can't see the forest for the trees.
Right. But if you compress it by 10x, the AI can process huge multi-protein complexes all at once. It finally sees the global picture.
And did they lose accuracy to get that 10x compression?
That's the best part. They maintained a similar distortion rate to the uncompressed models, so you get a much smaller file size with the same quality.
Okay, that's impressive.
But the real shock, and this is the part of the paper that genuinely made me sit up, was the data efficiency.
Let's talk about that, because we all know modern AI is incredibly data hungry. We practically feed it the entire internet just to teach it basic English grammar.
Right. And most protein language models out there are trained on millions of structures. Millions.
So what did GEOBPE use?
GEOBPE was trained on a data set of only about 34,000 structures.
That is nothing. I mean, in the AI world, that's a rounding error.
It's tiny. But here's the real kicker. They ran an experiment where they trained it on just 1% of that already tiny data set.
So what, a few hundred proteins?
Roughly, yeah. And it still matched the downstream accuracy of the massive models.
Wait, how is that even possible? How can an AI learn the complex rules of protein folding from a few hundred examples?
It shouldn't be possible, not if it were just memorizing statistical patterns like older models do.
Right.
This suggests that GEOBPE isn't memorizing at all. It's actively capturing the physics.
Because the tokenizer itself is built on geometric logic.
Exactly. Because it has the glue and the residues built into its foundation, it learns the rules of the game almost instantly. It knows how proteins are allowed to bend because the math of the tokenizer forces it to know.
It's not guessing based on past data. It's deriving the shape from first principles.
You nailed it. And the concrete proof of this came in their out-of-distribution tests.
The ultimate final exam for any AI.
Exactly. They took the model and showed it proteins from the Cameo and CASP14 data sets. These are brand new, highly unusual structures it has never seen before in its life.
How did the older models do?
The standard VQ-VAE models, the barcode ones, completely crumbled. Their error rates jumped by over 6.4 times.
Because they had no barcode for these new weird shapes, so they just panicked.
Right. And GEOBPE.
Let me guess. It held up.
It held incredibly steady. The error ratio stayed between 1.0 and 1.1. It barely even blinked.
That is wild. It proved that it had actually learned a language that could describe things it had never even encountered before.
Exactly. Which brings us perfectly to the other big promise of this paper, which is interpretability.
Yes. We talked about the black box problem earlier. Usually AI features are just abstract numbers in a matrix.
But since GEOBPE is building actual 3D shapes for its tokens, this becomes a glass box moment.
Because every token represents a real physical shape that we can actually look at.
Yes. We can look at token 452 and render it on a screen. And when the research team actually did this, they found that the AI had essentially rediscovered biology all on its own.
What do you mean rediscovered?
The tokens it created naturally aligned with concepts that human biologists have spent decades cataloging. Things like seed-cath domains. The AI independently found binding pockets and helix bundles without ever being prompted or told what those things were.
It found the functional words of biology just by looking at the geometry of the shapes.
It did. Let's look at the specific case studies they provide because they really drive this home for anyone listening.
Sure.
They analyzed a protein called SLC25A20.
Catchy name.
Very. It's a mitochondrial carrier protein. Its entire job is to physically shuttle molecules across a cellular membrane.
So it's a transport truck.
Right. Now, the model highlighted a specific sequence of tokens 48 through 52. When the researchers isolated that shape, it corresponded exactly to the transmembrane binding cavity.
So the AI pointed to a specific clump of geometry, said this right here is a meaningful unit, and that unit turned out to be the literal engine of the transport truck.
Exactly. It identified the specific cavity made of helices and polar residues that actually does the physical work. It didn't just see a shape, it saw the functional purpose of the shape.
That is incredible.
Yeah.
And there was another case study relating to neurodegenerative disease, right? The 14-3-3 complex.
Yes, the 14-3-3 tau complex. This is a huge deal for Alzheimer's research. The model identified a phosphobinding groove, which was token 29 through 33.
And what does that look like visually?
Visually, it's a very specific curved pocket lined with charged side chains, specifically arginine and lysine.
Which, chemically speaking, are very sticky.
Right, they are specifically designed by nature to catch and hold phosphorylated proteins. The AI saw that chemical stickiness just by looking at the geometric hierarchy of the shape.
So if I'm a researcher working on Alzheimer's, I don't just get a generic prediction score from this AI. I can look at the protein sequence and literally read these tokens.
Exactly. You can say, oh, there's a binding groove token, there's a transporter token. It makes the protein completely readable to a human.
It validates the idea that nature uses modular design principles. I mean, biologists have always suspected it, but now we have an algorithm that sees the microscopic world exactly that way too.
It's a huge paradigm shift for structural biology.
Okay, so we can clearly read biology better now. But the holy grail here, and let's be honest, the reason so much funding is pouring into this specific space is writing biology.
Intuitive AI.
Exactly. Can we use these newly discovered tokens to design brand new drugs or custom enzymes?
They tested this as well. It's an evaluation they call the Small Structure Language Model, or SSLM-EVAL.
What did they do?
They took these GEOBPE tokens and trained a small transformer, basically a mini version of GPT, to generate entirely new proteins from scratch. Unconditional generation.
Literally writing new biological stories with this new vocabulary.
And the results were fascinating, specifically regarding something called designability.
Which is a crucial metric, because you can ask an AI to draw a protein, and it might draw something that looks amazing on a screen, but violates the fundamental laws of thermodynamics.
Right. It creates an M.C. Escher painting. It looks cool, but it's impossible to actually build in reality.
We want actual blueprints, not fantasy art.
Exactly. And GEOBPE generated backbones that were 99% designable.
99%?
Yes. That means an inverse folding model could easily find a real sequence of amino acids that would fold into that exact shape in a physical test tube.
That seems incredibly high. How do the older barcode models compare in that test?
This is where the nuance really matters. The VQVAE models actually produced more diversity. They made wilder, crazier shapes, but they were far less designable. They were hallucinating structures that physically couldn't exist in reality.
So the older models were more creative, sure, but they were writing biological fiction. GEOBPE writes nonfiction.
That's the perfect way to put it.
Yeah?
GEOBPE produced realistic functional structures, long alpha helices, bundles that look exactly like real DNA-binding motifs or cytokine folds.
Because the system is entirely grounded in the physics of the glue and the residues we talked about.
Exactly. It is constrained by physical reality. It literally cannot hallucinate nonsense as easily because the mathematical rules of the tokenizer just won't let it.
Which is exactly what you want if you're a pharmaceutical company. You do not want to waste millions of dollars on a drug candidate that is physically impossible to manufacture.
Precisely. It strongly suggests that GEOBPE could be the foundation for a generative biology that is actually reliable. We could write code for a custom protein that, say, eats plastic and be 99% sure it will physically fold correctly when we synthesize it.
That is just mind-blowing. So let's zoom out for a second and look at the big picture for you listening. We started this deep dive with this grand idea of reading the language of life.
Right.
Feels like we've moved from trying to decipher a really blurry, noisy photocopy to suddenly having the crisp digital source code.
I think that's a very fair summary. GEOBPE is fundamentally bridging the gap between the discrete world of computer science, which absolutely loves tokens and tidy lists, and the continuous, messy world of physics, which is all about fluid curves and dynamic forces.
By using the glue to fix the spatial math and the clustering to find the common vocabulary.
Yes. It essentially translates physics into a language computers can natively speak.
And crucially, it solves the black box problem that plagues so much of AI right now. It pushes us away from these mysterious, arbitrary computer vectors and towards highly interpretable visual motifs.
It lets us actually see the Lego blocks of life.
It lets us see how nature builds complexity from simple modules. And that alignment is key. When our computational tools finally align with the physical reality of the universe, that's usually when we make the really big historical leaps.
Absolutely.
It really makes you think, though. If this team from MIT and Apple could rediscover the functional words of protein biology just by analyzing the geometry and the math, what else is hiding in plain sight out there?
You know, that is the exact provocative thought I've been mulling over since I first read this paper. Nature is completely full of complex, continuous systems.
Weather patterns, the flow of fluids in an engine.
Tectonic shifts, even human-made systems like the stock market. We usually treat these things as just continuous, unpredictable chaos.
But maybe they have a distinct language, too.
Exactly. If an algorithm can find the literal vocabulary of protein folds just by looking at structural frequency and geometry, what other hidden languages exist in nature that we simply haven't built the right tokenizer for yet?
What if we could tokenize the weather?
Cloud pair encoding.
I mean, why not? Finding the underlying grammar of the universe might just be a matter of finding the right mathematical way to chop it up.
That is definitely a thought to keep you up at night.
It really is. As always, the links to the full paper and all the sources are in the show notes if you want to geek out on the inverse kinematics and the math yourself.
It's highly recommended. It's a great read.
Thanks for diving in with us today. We'll see you on the next one.
Take care, everyone.